In the previous video, we talked about what lipids are, what are their functions, and their properties. This next video will discuss what are the different classes of lipids and how do we classify lipids. So lipids can be categorized under two categories. First is saponifiable lipids, and second one is non-saponifiable lipids. Saponifiable lipids will be the lipids which contain ester bond, and that ester bond can undergo the process of saponification. Whereas non-saponifiable lipids will be lipids that do not contain any ester bond and therefore they will not undergo any saponification. Saponifiable lipids have the first two classes under them, which is simple lipids, and the second class is compound lipids. Compound lipids or complex lipids. Non-saponifiable lipids have third class of lipids called derived lipids. So these three classes, simple lipids, compound lipids, and derived lipids, their name suggest what these lipids will be like. Simple lipids will have a simple structure, compound lipids will have a relatively complex structure, and the derived lipids will be the lipids which can be derived from the first class of lipids and the second class of lipids. So these are three classes. Now let's talk about these three classes in a little bit more detail. First class of lipids, simple lipids, is simple in structure, which means it has very few components, and the main components of simple lipids are alcohol plus fatty acids. So this is a simple structure or a general structure of a simple lipid. Simple lipids have the property of being non-polar in nature, and simple lipids are useful for generating ATP. That's why we also call them storage lipids, because they are stored in our bodies for generation of ATP. On the basis of what kind of alcohol is there in the simple lipids, we have two types. First is triglycerides, also called EAG, also called neutral fat. The second type is wax. Triglycerides have glycerol as alcohol, and glycerol has only 3 carbons. In case of wax, the alcohol can be any alcohol, but it should not be glycerol. And the alcohol in case of waxes are very long chained. The alcohol can have from around 14 carbon to 30 carbon. The second component of simple lipids, fatty acids, are present again in both these types. Fatty acids are present in triglycerides. Fatty acids are present in waxes as well. But the fatty acid of waxes are again very long chain. They can have carbons ranging from 14 carbon to 36 carbons. Let's have a look at the general structure of glycerol. Glycerol has only 3 carbons, so there are only 3 sites where fatty acids can go and attach. Fatty acid can attach here, here, and here. The linkage between glycerol and fatty acid is ester linkage. And that's why sometimes we call glycerides as fatty acid esters of glycerol. And depending upon what kind of fatty acid is there, whether the fatty acid is long chain or short chain, or whether the fatty acid is saturated or unsaturated, the whole property of that triglyceride will change. Few examples of triglycerides are the vegetable oils we use for cooking, ghee, butter, etc. Few examples of waxes are beeswax, earwax, carnauba wax, lanolin, etc. Beeswax, carnauba wax, lanolin are used in pharmaceutical industry. They are used in preparation of cosmetic products. Earwax, which is present in our ears, helps in trapping of dirt particles. So this was the first class of lipids, simple lipids. The second class of lipids is compound lipids, also called complex lipids. Compound lipids will have these components. There will be alcohol, fatty acid, and attached to them there are different conjugate groups. Depending upon what kind of conjugate group is attached to alcohol and fatty acid, compound lipids are of three types. First is phospholipid, second is glycolipid, third is lipoprotein. So their names suggest what components or what conjugate groups are attached. In phospholipid, the conjugate group that is present is phosphate. Now this phosphate molecule is going to give polarity to the phospholipid. So this gives polarity. And one more property is that phosphate is reactive in nature, so it will tend to attach to some other group. And that other group, we are going to assume that it is an X group, which is going to change the property of that particular phospholipid. Now again, depending upon what alcohol is there in phospholipids are of two types. First type is glycerophospholipid and second type is sphingophospholipid. If the alcohol is glycerol, then phospholipid will be called glycerophospholipid. If the alcohol is sphingosine, which is an amino alcohol, then the phospholipid will be called sphingophospholipid. Glycerophospholipid will have two fatty acids attached to it. They can be same or different. And along with that, at the third position, there will be a phosphate group. Because the phosphate group is reactive, phosphate group is going to again react and attach itself with the next group. For sphingophospholipid has alcohol sphingosine. And this sphingosine is very large in size. To the sphingosine, only one fatty acid is going to attach. And at the second position, phosphate group is going to be attached. And to this phosphate group, some X conjugate group will attach. Examples of glycerophospholipids are cardiolipin, lecithin, and cephalin. And one common example of sphingophospholipid is sphingomyelin. So this was phospholipid. All the lipid molecules to which phosphate group is attached will fall under the phospholipid category. The second type of conjugate lipid or complex lipid or compound lipid is glycolipid. Glycolipid will have lipid molecule, which means there will be alcohol, there will be fatty acid, and to this will be carbohydrate attachment. This carbohydrate can be monocarbohydrate, monosaccharide, disaccharide, or oligosaccharide. And the alcohol is not glycerol. Alcohol is always going to be sphingosine. So in case of glycolipids, the alcohol is going to be fixed. Sphingosine amino alcohol is going to be used. Glycolipids further can be divided into three different types. These are cerebrosides, globosides, and gangliosides. Out of these three, the first two are non-polar, and the third one is polar. We'll have a look at their structures in coming videos. The last type of complex lipid is lipoprotein, and lipoprotein will have lipid component and protein component. Lipoproteins are different from the first two categories of complex lipids, so we'll just have a look at their examples right now. Chylomicrons help in process of digestion of lipids. VLDL is very low density lipoprotein. LDL, low density lipoprotein. HDL, high density lipoprotein. So these are the examples of lipoproteins. This was the second class of lipids, the compound lipids, and their three subtypes. The third class of lipids is derived lipids. And as the name suggests, the derived lipids are going to be derived from first class of lipids or second class of lipids, or they can be derived from other derived lipids. So we are just going to have a look at the different examples of derived lipids. These examples fall under three broad categories. First category is sterols. Second category is eicosanoids. And third category is isoprenoids. Few examples under these categories. Sterols are used in synthesis of cholesterol. Cholesterol further is used in synthesis of several hormones and vitamins. Hormones like progesterone, estradiol, aldosterone, cortisol are synthesized from cholesterol and vitamin D as well. Eicosanoids is a group of three specific compounds or chemicals: prostaglandins, leukotrienes, and thromboxanes. All these three chemicals, prostaglandins, leukotrienes, and thromboxanes, they are useful in intracellular communication. Isoprenoids again contains a vast list of chemicals like vitamin A, pigments like carotenoids, terpenes like essential oils from different plants, pine oil, lime oil, geranium oil, etc., rubber, and phytol ring, chlorophyll, etc. One more compound that I want to add is bile salts. They are also derived from cholesterol. So this was a list of different chemicals under the categories of sterols, eicosanoids, and isoprenoids. Now, summing up the classification of lipids, we can say that lipids have two categories called saponifiable and non-saponifiable lipids. Under saponifiable lipids, further have two categories: simple lipids and compound or complex lipids. Under non-saponifiable lipids, we have the third class or third category called derived lipids. Under simple lipids, on the basis of alcohol, we have two types. First is triacylglycerol or triglycerides, also called TAGs, and second one is waxes. Under complex lipids, we have three different types. First is phospholipid, then is glycolipid, and third one is lipoprotein. And derived lipids will have three categories or three set of categories: sterols, steroids, and isoprenoids. Under phospholipids, we further studied two different types of phospholipids: glycerophospholipid and sphingophospholipid. Under glycolipids, we studied cerebroside, globoside, and ganglioside. So this is a general flowchart that you can use for your exams or use for learning how lipids are being classified. So here I end today's discussion on classification of lipids. In the next video, I will talk about each of these different classes in detail.